Hello everyone, I will be telling you about a recently published paper, the integration and harmonization of trade data from plant individuals across heterogeneous sources, which has been published in the Journal of Ecological Informatics. This presentation will consist of two main parts. First, I will show you the workflow which can be used for the automated integration of plant traits. And second, I will show you about an integrated palm trait dataset which may be useful for your own research. So as a brief overview, 4 parts. The first is I will explain what the current challenges are for palm trait research. Second, how does our workflow aid in the automated integration of trade data and how does it help to tackle some of these challenges? Third, I will show you a palm trait dataset which we mobilized containing nearly 140,000 individual trade measurements. And last, I will show you where you can find additional information and documentation if you are interested in using this method. So, in recent years, we've seen a sharp increase in trade-based research in ecology. This is not so strange as traits are critical for the understanding of plant form and function and have shown to be relevant for the ecosystem population and ecosystem functioning. One important aspect within trait research is interest-specific trait variation. This has been shown to be relevant for fields like global change, biodiversity change, and policy research. The problem is that there's currently a limited availability of individual trait measurements. This is mostly because current efforts that seek to mobilize these trait measurements aggregate them at the species level, with the result that this interest-specific variability is not captured. Therefore, this variability is only often available from personal data storages and herbarium collections, which are often not accessible or even not digitized. In short, these records do not follow the FAIR principles, meaning that they're not findable, accessible, interoperable, and reusable. So how did we try to resolve this? We created this workflow for the integration of plant trait data. We illustrate how this workflow works by using palm data. Palms are a pantropical plant family with a very large trait variability and therefore very suitable to use in this workflow. We take unstandardized trait measurement spreadsheets and provide a standardized integrated data product, which does follow the FAIR principles. It also follows the ecological trait data standard, which means that the final data product consists of 4 tables, a core tables holding all the measurements and 3 extension tables, which provide additional information. It also follows semantic standards from the ecological trait test standards, Darwin core, plant trait ontology, and the thesaurus of plant characteristics. So our workflow consists of 3 main steps. First, the input. The data provider, which could be someone who has these personal data storages of trait measurements or is seeking to make an integrated data set. It provides trait measurements in Excel spreadsheets. He then uses metadata forms where he uses 1 form per trait measurement spreadsheet and he enters the column headers from those spreadsheets into the metadata forms. These column headers are then automatically linked to ontologies for different ones for traits and for other metadata. These collection of metadata forms are then integrated into 2 different thesauri, namely the metadata thesaurus and the units thesaurus for the measurement units. These thesauri are together with the raw spreadsheets are used as an input in the R integration workflow, where the spreadsheets are formatted, integrated through column header standardization. They're converted from a wide to a long table format. And additionally, there are some standardization efforts such as taxonomic standardization and providing additional information, as well as coordinates standardization. This will feed into the outputs, which consists of 4 tables. First is the core table, consisting of a species name, per row of a species name, a trait value, and a trait unit, as well as a trait ID. So this is then linked through these ontologies. Additional information about these measurements can be found in the 3 extension tables below, and they are linked through these ID columns. As an example, what kind of data sources can be integrated? Here we have 2 small parts of these trait measurement spreadsheets. As we can see here, one row gives all the trait values from one specific specimen. For instance, if we want to integrate the values for stem length, in this data frame, this is this column header, whereas in the other one, it's a different column header. In this case, this is actually an easy example because you can pretty easily derive still what it means. But this is kind of a best case scenarios as we have encountered, as a lot of these column headers are often not as descriptive. Another example is the stem width, which are also both different column headers in these data sets. Now I will show you how the metadata form works. So first, the dataset provider will give some information about his name, so the name of the dataset provider, what the date is, the dataset name, the reference, if there's a publication linked to this dataset, additional spatial information, and also the basis of record. He then matches column names for traits to standardized names, which are provided in this metadata form. If the user is not sure what is meant by one of these standardized names, they can be clicked, and they will, because they're linked to ontologies, will send you to a web page with extra information about this trait name. Other than the column header for the trait name, he will also provide the measurement unit. This is the same for the extension tables, where these are also column headers, but then not for non-trait columns. As explained, these feed into these 2 different thesauri. So here we have an example of 2 metadata forms from 2 different data sets. And as we see here, the column headers are fed into the metadata thesaurus, where one column is used for one metadata form, and the same is for, and this is the same for the units. So we did this for 15 different data sets, palm trait data sets, and we mobilized almost 140,000 individual trait measurements. This data set contained 50 different standardized trait names and 551 unique palm species, which is roughly 22% of the total known palm species globally. And this is what the core table looks like. So you will have your scientific name, the verbatim trait name, so this is how it is used in the original spreadsheets, the standardized trait name, trait value, and trait unit. These are all converted and standardized. And then a trait ID, which is the URI which links to the trait name. And then the 3 different columns relating to the extension tables, which are the taxon extension. We will give you additional taxonomic information about each species. This information is derived from GBIF, the plant list, and the World Checklist for Vascular Plants. And they have a coverage of 78 to 89%. The occurrence extension will give you additional information of where the specimen has occurred, for instance. And we see here that in this data set, we have specimens for 60 different countries, which are also globally distributed. Finally, the measurement or fact extension contains over 3,000 combinations of additional information related to the measurements itself. And then I will briefly show you some additional files which you can use, or download links if you are interested in this workflow. So first, this is the web page for the article. So in this article, we will really explain to you in depth what the different, how this workflow works, and also additional information about the palm trade dataset. Second is the trade data GitHub repository, which holds all the files needed if you want to use this workflow. It also provides documentation to show you how this works. If you're interested in using the R-based workflow, I would suggest downloading the entire repository, as these folder structure works seamlessly with the workflow, and you wouldn't have to change any inputs. You'd only add your own spreadsheets to the raw datasets folder. Third is we also provide a data dryas repository. If you're only interested in the palm trade dataset, that can be downloaded here. And last, we've made a bookdown tutorial, which will give you an in-depth tutorial of all the steps if you want to use or actually alter this workflow. This workflow is open source, so any functionalities can be added. Anything that doesn't work for you can be altered. So in short, what does this consist of? First, an explanation how to use the metadata form. Second, how do I move the information from this metadata form to both of the thesauri? The third is for every step of the R script, an explanation, what does this step do? And also, if you want to add something, you can see where you have to do that here. And fourth, I will show you some example outputs of the output tables of this workflow, which will also give you explanations of how to use it and how to read those. With that, I'd like to thank you for watching. All the links to these web pages are put in the description. I'd like to thank the Palm Symposium 2021 for providing me with this opportunity. And yes, thank you.